Good evening. I am David Campbell, University Provost, and on behalf of all of us here at Boston University, it's my great pleasure to welcome you, all of you, full crowd, to this 2008 University Lecture. Established in 1950, the University Lecture provides an opportunity for us to learn from a distinguished member of the Boston University faculty who is actively engaged in outstanding research or scholarship. It is a forum open to all, students, faculty, and staff at the university as well as to our neighbors in the greater Boston area, and that one that involves in considerations of issues that are important to our understanding of the world around us. Over the decades, these lectures have galvanized discussion, provoked inquiry, and informed us about topics as diverse as language acquisition, presidential disability, genomics, the atmospheres of distant planets, and the current relevance of the theologian Reinhold Niebuhr. Despite continuing trends toward interdisciplinary research and collaboration, it remains rare, even in academia, to hear a lecture from someone with expertise in a realm entirely different from our own. The beauty of the university lecture is that all of us, humanists, social scientists, natural scientists, engineers, medical practitioners, and others, come together to learn, to be inspired, and to open our eyes to a wider world. As a former Dean of Engineering, I take particular pleasure in offering you tonight the insights of one of our most distinguished biomedical engineering colleagues. To introduce that colleague and his topic for the evening, I am very pleased to ask Bob Brown, Boston University's 10th president, to come to the microphone. Please join me in welcoming President Brown.
Good evening. It's wonderful to see a full house. We are very proud tonight to present Dr. James J. Collins, a university professor, professor of biomedical engineering, and co-director of the Center for BioDynamics. Jim's lecture is entitled Biology by Design. Professor Collins has been a member of our faculty since 1990. He graduated summa cum laude in physics from the College of the Holy Cross, where he was class valedictorian, and earned his doctorate in medical engineering from Oxford, where he was a Rhodes Scholar. Jim is the recipient of numerous awards and honors for his scientific work and for his teaching, including the Metcalf Cup and Prize, the university's highest award for teaching excellence. He is the first biomedical engineer to become a recipient of a MacArthur Foundation Award. And this past year, he was named a Howard Hughes Medical Institute investigator, the first from Boston University. Jim is a wonderful and complete researcher. He pursues his curiosity through basic research, and he finds exciting and practical applications for his findings. His research focuses on developing nonlinear dynamical techniques and devices to characterize, improve, and mimic biological function. He is a pioneer in a newly created field of synthetic biology, the design and synthesis of biological systems that do not already exist in the natural world. He holds many patents and has co-founded 2 companies based on his technologies, and I'm sure there'll be many more to follow. What is less known about Jim is his talent as a southpaw. On Friday, August 29th, he was invited to throw out the first pitch at Fenway Park. The Red Sox beat Chicago that evening 8-0. I believe it's fair to say that if Terry Francona had started Collins in Game 4 of the ACLS, we may have had a different outcome and a different evening tomorrow night. Now, it is no stretch for me, as it was to talk about his pitching prowess, to welcome him as one of our most distinguished members of our faculty to present his lecture. Dr. Collins.
Well, I want to thank President Brown for the really nice introduction. And I want to thank all of you for coming out tonight. It's really great to see so many young and old faces in the audience. What I want to do tonight is tell you a bit about what our lab has been doing since I joined Boston University. And really, I want to tell you about 3 different areas. One is our work on using noise to enhance human sensory function. Second is to tell you about our work in synthetic biology. And third, to tell you about our recent work in systems biology pointing towards the development of new antibiotics. So first, this strange notion of using noise to enhance human sensory function. Where it's strange is that we generally don't think about noise as being beneficial for enhancing the ability of someone or something to detect a signal and to transmit information. Now here, when I refer to noise, I don't mean a loud, annoying sound like my voice. Instead, I mean some randomly fluctuating signal, which may be electrical or mechanical in nature. In general, we try to get rid of this noise if we're trying to detect a signal or transmit information. So we have engineering textbooks and whole courses dedicated to training students to get rid of that noise. So why would you ever think about introducing it into something? Well, the story goes back almost 30 years ago now to two independent groups of theoretical physicists who were doing modeling of the Earth's climate. And what they were trying to do was account for the periodic recurrences of the Earth's ice ages. So about every 100,000 years or so, we have an ice age. We're overdue for an ice age. We're expecting one. What these physicists did was consider a very simple model, basically a two-well potential, where you have a marble representing the state of the Earth, where on one side you have an ice age, the other side you don't have an ice age. You then have a barrier separating those two wells. And they considered some periodic geophysical force that could attempt to flip the Earth's state between ice age and no ice age. They found that this was a sub-threshold signal, so that in general it was not detectable. It wouldn't flip the Earth between those two states. But if they added a little bit of noise, fluctuations in some sort of geophysical variable or parameter, they could actually occasionally cause it to flip, hence accounting for the periodic recurrences. Okay, pretty funky, weird notion. Putting that aside for a second to give you an appreciation for what this model really means, imagine instead that I'm standing in front of you and I have, say, a two-well egg carton. Okay? And let's imagine further there's one marble in one of the wells. And I'm going to move my hands back and forth periodically. Okay? Let's assume further you can't see my hands, and you want to try to figure out what are my hands doing. Okay? Let's now assume further that I'm moving my hands with an amplitude that doesn't cause the marble to jump from one well to the other. Okay? Now go further and say, okay, you can't see the marble if it's in one well. All you can see is if a marble makes a jump from one side to the other. If I'm standing here in that situation, moving my hands back and forth, it's a sub-threshold situation. You can't tell me anything about what my hand is doing. Okay, so you can't detect the signal. Now let's imagine that I stand on the tee outside. We're on the B line. And let's say we have my daughter, Katie, who, like me, is physically averse driving the tee. She's physically risk averse, so she's going to drive the tee slowly. Right? So if you're on the tee, you're going to get random vibrations. Right? So at slow speed, you're going to have a little bit of noise now coming through your body. And if I'm still moving my hands back and forth periodically, every once in a while, the marble is going to basically now make a jump from one side to the other as a result of the interaction of the vibrations now, the noise, with the movements of my hands. So if you watch it long enough, you can say, gee, I think Jim is actually moving his hands periodically. Let's say you go on the other extreme. Now, say my wife, Mary, is driving the tee. She likes to drive fast, as any state troopers in the audience can attest. And so if you drive the tee quickly at a high speed, you're going to have large amounts of vibration, right? So now if I'm moving my hands back and forth periodically, now the marble's going to jump a lot. Why? Because I'm being vibrated at a high amount. Okay, so now if you watch the marble, you're not going to get much information about what my hands are doing. It's instead going to be dominated by the noise that's getting transmitted through my skeleton, kind of more classically the way we think about noise. Now let's kind of go into me. Let's take my son Danny, who is kind of a house blend. He likes speed like my wife, but he'll slow down if you ask him to. So say he drives a T now at an intermediate speed. Now you'll have more noise in the low speed, less noise in the high speed. And it'll be basically a Goldilocks level. So now the noise is going to come through and add more or less constructively with the movements of my hands, so that now many of the leaps of the marble will actually be correlated with the movements of my hands. So you can actually, he's moving it periodically. It's, say, once every second, he's moving his hand up and down. That, in essence, is stochastic resonance. This notion you can introduce noise into a system and enhance the ability to detect previously undetected signals. So if you look at a plot where you're looking at the amount of noise, and you're looking at some measure of information, that is, in this case, the input-output coherence. What can you say about the movements of my hands? When there's no noise and it's a subthreshold signal, you have no information. At low noise, when Katie's driving the T, you have a little bit of information, but not a lot. At high noise, when Mary's driving the T, a little bit of information, but not a lot. And then when Danny, at the intermediate Goldilocks, you peak. And that, in a nutshell, is stochastic resonance. Stochastic is just another term for noise or random. And resonance is a phenomenon that's studied in physics and engineering where you introduce and vary some input, and you get some peaked output as you vary it. This has been studied in hundreds of papers. Some actually leading experts of the space are actually in the audience today. largely in the physics literature. Frank Moss, a physicist at the University of Missouri at St. Louis, in the early 90s, identified stochastic resonance in crayfish, of all things. He found that if you looked at mechanically sensitive neurons in the tails of crayfish, that he could actually see evidence for stochastic resonance. Specifically, that you would have weak, say, predator movements in whatever crayfish live in, some water body, that it could better detect, as a result of the noise, those weak movements. I met Frank at a NATO workshop in February '94 outside Montreal. He knew I was in a biomedical engineering department. So he pulled me aside and he says, Jim, if you can come up with a medical application for stochastic resonance, you'll never have to write a research grant again. The NIH will drive a dump truck of money up to your lab every October. You'll be home free. Well, there are many academics here, as I know, like all of you. I don't like to write grant applications. But Boston University, in particular, a lot of the guys sitting here, like it when I get grant money. So with suitable motivation, I basically, on the flight home, began to think about, well, what could you use this for? And as engineers, what we can do is we can artificially introduce noise into the body, either in the form of mechanical vibration or the form of electrical noise. And I realized, well, crayfish have neurons. We have neurons. All of our sensory neurons are threshold-based. So you have a barrier like we had in that 2L potential in the first slide. Right? So if I have you close your eyes and I press on your fingertip, I have to press on your fingertip with some amount of load before you sense I'm pressing on your fingertip. So for a young, healthy person, it might be half a gram of load. Okay? If you get older, as you hit about 65, your thresholds start to go way up. So about 65, instead of half a gram of load, now it might be 5 grams of load. This poses significant difficulty for the elderly to interact with the environment. They can't feel as well, and they lose sensation in their legs and their feet. And as a result, they lose the ability to maintain balance. So as many of you know, falling is a huge problem for the elderly. 1 out of 3 individuals over the age of 65 fall each year, and many of these falls result in serious injury. They end up having to be hospitalized. Many will lose their independence. If you talk to older people, survey them, their greatest fear is not dying, it's falling. So we thought, well, this would be cool. Could we maybe design vibrating insoles on the basis of stochastic resonance that we could give to Mrs. McGillicuddy so that she could better balance and walk around and do her stuff? Further, in addition to aging, what also happens is that with disease, thresholds will go up. So in the case of diabetes, if you leave the disease untreated, you could develop neuropathy, where you lose sensation in your hands and feet. And as a result now of the neuropathy, particularly in older subjects, instead of now 5 grams of load as a 65-year-old, 10 grams of load. Huge problem in terms of sensation, movement, but as well as detecting things in your feet, in your shoes. So in a diabetic, there might be a pebble in their shoe if they have significant neuropathy. That pebble can basically rub away, lead to a small cut, and lead to an infection, and in the worst case, lead to an amputation. Sounds extreme, but in fact, in this country alone, there are over 50,000 amputations performed on diabetics each year. So we thought, wouldn't it be if you could maybe use noisy socks, using electrical noise they could put, or noisy gloves to help diabetics overcome neuropathy. Third is stroke. It's basically a central disorder. In stroke, you'll basically have blood blocked from going to your brain, shut down part of your brain, and as a result of it, you actually lose sensation. So now instead of 10 grams of load needed at your fingertip, it might be 20 grams of load, often resulting in paralysis. So we thought, okay, maybe we could actually develop clothing type equipment that could be used in rehab, both to restore function on every day as well as through rehab. So we got really excited. I came back to the lab, and with Carson Chow, who was one of my postdocs, we kind of sat down and said, okay, what could we do fast? Well, we went to where we usually go in engineering, as we first started doing computer modeling of neurons. Why? It's cheap. You don't have to feed computer models of neurons. 2, it's fast. I think there's some people from the IRB. I don't have to get ethical approval to do experiments on computer models of neurons. And what we did was model these and then introduce. We looked at a number of different neurons, and with Carson Chow, who's a theoretical physicist, we basically studied these on a computer and actually found for broader. So the physicists up to that time had only studied periodic inputs. But as all of you know, we actually interact more than just periodically, right? Mrs. McGillicuddy doesn't just kind of dance around always to the beat. She moves around kind of aperiodically. And we were able to show in our neurons that as you increase the input noise, just as you saw earlier, broadly as the physicist demonstrated, the amount of information detected by the neuron, the model neuron, increased to peak and then went down. So that there was some level of noise in particular where you could maximize the amount of information your neuron picked up. Well, I can go on. 50 minutes. There are several mathematicians. Some of the world's leading biomathematicians are here tonight. I can give 50 minutes of talks. You don't want to hear that. The biologists don't want to hear it. Clinicians don't want to hear that, and we realized that. And we realized in order to have an impact, we had to go beyond model neurons. So we set up a collaboration with Peter Grigg at UMass Medical Center and actually moved to do animal experiments. So what we did in these experiments is we took rats and actually looked at small patches of skin in the rats where we could maintain sensory neurons and delivered small mechanical signals that the neurons could not detect. Okay? We then introduced noise of different amplitudes. And in all cases where we could get the data, and it's another story, maybe questions, we could actually get stochastic resonance type behavior. So as we increased the input noise on the rat neuron, the ability of that rat neuron to detect weak signals increased to peak and then slowly decayed, showing that we could actually now enable a rat neuron to go get weaker signals than they normally could and designed to. Really neat result moving from computer models now to actually real rat neurons. Well, I have many career goals, as alluded to by President Brown. One of them is not enhancing sensory function in rats. When I die, I don't want my obituary to say the rat community is devastated by the loss of Professor Collins. I do have interest in enhancing sensory function in humans. So we moved up the food chain and came back to here to BU and did experiments on BU undergraduates who we knew were at least physically healthy. So with IRB approval, and this was not one of the subjects. This is actually Attila Priplata, one of my star PhD students who helped do the studies. We basically took the indenture that we used on the rat skin and now used it on the undergrads. Oh, and used it to, sorry, I've got to get used to this clicker. We used it to introduce weak signals on the fingertips of the subjects. Okay? Signals they previously could not detect. So here's now 20-year-olds, very healthy 20-year-olds. And what we were able to show was as you increase the input noise on these young undergraduates, their ability to detect previously undetected signals increased to peak and then decreased. So that there were some levels of noise where they could go and get signals they previously couldn't feel. What was great about that result is you generally think of 20-year-olds kind of optimized. We all hark back to wish we were 20 years old again. Our bodies work great. But in fact, we show that they're not optimized. There still is room to improve. So we could go and get them to go get weaker, smaller signals. With the same setup, we actually were able to take 65-year-olds, 75-year-olds, improve their sensory function, bring them down to the level of 20-year-olds, as well as we could do it on diabetics both on fingertip and feet and improve sensory function, as well as take stroke patients and similarly introduce the noise and reduce their sensory threshold so they could also feel better and close to basically 20-year-olds. So the big question then after going from here is, well, that's fine on touch sensation. Could it actually enhance sensory function in Mrs. McGillicuddy? So could you use your vibrating skateboard, in this case, to enhance the way that Mrs. McGillicuddy might be able to maintain balance? So with Attila Priplata and Jim Nemi, who were working in our lab, what we did is we got in the lab, and basically what the guys designed was essentially a vibrating shoebox. So took a large metal plate, introduced hundreds of holes, small holes in the top of the plate. Underneath the plate, we introduced two motors that had attached small plastic pylons that came up through the holes in the plate and made contact with the subject's feet. Okay? It could then deliver random vibrations. So in our lab at the time, you could get a very high-priced foot massage if you were interested. We actually would set up the noise so you couldn't feel it. So now instead of showing you results, we're going to sweep the noise. What we found was, that, interestingly, that the peaks in the subjects, human subjects in particular, where you saw the maximum amount of information being boosted by the noise, occurred just below their ability to feel the noise. So you could deliver noise they couldn't feel to get the beneficial effect that we were seeking. So what we do in the lab is we then can put reflective markers on different parts of the subject's body and use television cameras that can record how those markers are moving. So in each of you, if you try to stand still, you can't do it. We actually all sway a bit. And the amount of sway is reflective of how good your balance control system is. This is kind of a form of motion capture. So again, for the kind of the young, I see several teenagers out there, this is used to basically make Madden NFL football. You basically wire people up, collect the movement, and then plug it into your computer. Here, we use it actually from a biomedical standpoint. We can record to see how well do they maintain their balance when we deliver the noise versus when we don't deliver the noise. When we do that, I won't go through this plot in detail because you'll fall asleep post-dinner, is we basically looked at a number of measures of how well you maintain balance and found that when we introduced the noise, all of those measures improved. So they maintained better balance with the noise. Many of them were actually statistically significant for very small sets of population. We looked at young subjects in their mid-20s, and we looked at elderly subjects from mid-60s to mid-80s. Most interesting, when we pulled out and looked at the elderly separately, we found that we could take 75-year-old Mrs. McGillicuddy and make her balance as well as 20-year-old grad student or undergrad at BU. Now, Mrs. McGillicuddy can't hop around with a huge vibrating shoebox around Boston, around Newton. So the next challenge is, could you actually introduce insoles to do the same thing? And what we did with Attila Priplata was design gel-based insoles. So these are actually based on Attila's size 13 double E foot and shoe. He introduces in these gel-based schemes, they're about an inch thick. He has tactors. These are small motors about the diameter of a US quarter and the thickness of several of those quarters. Two were placed underneath the balls of the feet, one underneath the heel. And this could be used to deliver subsensory random vibration on the soles of subjects' feet. Firstly, good luck getting these through airport security, but that's another issue. We take these, repeat now the protocol where we're going to have them stand on it, measure how they're balancing using our motion capture. And what we found was even better than what we had with the vibrating shoebox. And that is that with the young versus elderly, we found basically that all of the measures improved substantially. We again could get 75-year-old Mrs. McGillicuddy and have her balance as well as a 20-year-old. And interestingly, we also found that the elderly improved a lot more than the young, which is speculation we had. We go back and talk about when I said that we think of the young generally as being optimal. Well, there is a gap for improvement. And we had speculated that the elderly, that gap gets bigger. So if you have a bigger gap, then you should have more room to improve. And we, in fact, found that could happen. Now, there are a lot of challenges with Attila's insoles. You go through it. They're about an inch thick. So they might fit in Uggs. I know several people here wear Uggs. Most other shoes, they won't fit. What we were able to do was design a new prototype insole that was only about a ½ centimeter in thickness, a type like a Dr. Scholl's insole you might get at CVS. That's one. Two is the original insoles were very expensive. Each of those tactors cost about $300. So if you're going to get a pair, you're now looking at least at $2,000 for a pair. Some of you may be willing to pay that for shoes, or maybe you would have before the markets collapsed, but most of us would not. And one of the challenges for those who track medical technology is the real need to develop affordable devices. There's really cool stuff we can develop. Most of the stuff actually doesn't leave the lab because it's often too expensive. We were able to replace those motors with small little DC motors that instead of costing each $300, only cost about $1 to $2. The bigger challenge for those of you who actually are device is power. The original guys, these big tactors, basically required a car battery and last for about 2 hours. Infeasible, right? You can't have Ms. McGillicuddy with a car battery strapped to her back. Right? She's, hey, I'm at Cambridge Side Gallery, AAA. I need a jump. My shoes died. Right? It would be a disaster, right? Well, with this new design, we were able to actually drive these off of 4 AA batteries. And when we ran the experiments on these, they worked brilliantly just as well as the earlier versions. We've since developed funky, more space-like looking ones with piezoelectric insoles that is now being developed. And where is this going? Well, the technology's actually been licensed out to Afrin Corporation originally, which is a small BU startup that Bob had made reference to out in Providence, Rhode Island. Their general technology was just acquired last month by Stryker Corporation, which is a very large, multi-billion-dollar medical device company. They want to use it now to create a new platform in neurostimulation. And the insoles are actually being spun out into a separate company that's going to focus on the insoles and footwear technology. So with this kind of first example, what I wanted to give you a sense was how an academic researcher or an academic research team can go from global climate modeling to computer models of neurons, to rat studies, to human perceptual studies, to pre-prototype studies, to prototype studies, to commercialization, to actually now being picked up by a large company. It's an interesting set of transitions. The more interesting transition is how do you go from this, where you're studying whole body dynamics, to synthetic biology, basically studying DNA and engineering systems. Well, this story goes back to the mid-90s. This is Charles Cantor, who was the chair of our department at the time. He's a real maverick, unlike he's mavericky. Charles really was one of the pioneers in the Human Genome Project. And when I first joined Boston University, I was on research faculty, which had the downside for me that I didn't teach, but the upside that I never had to go to a faculty meeting. And at one of these faculty meetings, our department decided that they were going to apply for a $5 million grant from the Whitaker Foundation. This was a philanthropic group that was giving large grants to biomedical groups, biomedical departments in particular. And our department pitched to them that they wanted to train the next generation of BME students to go from genes to cells to tissues to organs to whole body. The department put in a pretty good proposal. They were going to be site visited. Now, the Whitaker liked because they now sunshine clause themselves out. They liked to see groups already kind of doing what they were pitching and proposing to do. Charles Cantor called me in his office. He says, Jim, we're going to be a little embarrassed. We don't have anybody kind of bridging these gaps. I know you're doing control theory, interesting nonlinear dynamics for the whole body. Would you consider doing something down at the level of genes and cells, proteins? I said, sure. What do you got? He says, I got this system. We've engineered this cell suicide system. It gobbles up stuff in the environment, and then it kills itself. But it doesn't work so well. It's really leaky. It's killing itself before it should. Could you model it and try to get a better control scheme going? I said, oh, sure. We don't know much at all, if anything, about molecular biology. We'll give it a shot. So Charles ran to his whiteboard, and he put up all these equations. I'm all excited. I'm writing them down. I said, okay, we'll do it, Charles. And as I'm leaving his office, he says, oh, Jim, stop. You have 4 days to come up with something. Classic Charles. He says, the site visit is on Friday. So we go back At the lab, we start playing with the equations. Again, there are a lot of techies in the audience. These equations were not interesting. There was no interesting dynamics. It turned out they actually weren't relevant to the system in the way that we had hoped. We didn't know enough molecular biology to actually do anything about it, make the adjustments. So I went back to Charles on Thursday night, and I said, Charles, I'm sorry. We came up with nothing. We're going to have to punt. And he says, no. I want you to go out there tomorrow and tell them your vision for what an engineer can do in molecular biology. So I went out the next day and gave my 96-hour half-baked notion of what an engineer can do in molecular biology. And we did not get that grant. Thanks in part to my presentation. Now, just shifting ahead a few years, Ken Luchin, who's now our Dean of Engineering, became the chair of our department. And Ken had the guts to go back to the Whitaker Foundation and say, hey, we want to try again. And this time, we don't want $5 million. We want $14 million. So Ken puts in a really strong proposal. And we're site visited again. And Ken comes down and says, Jim, I want you to present. I says, really? He says, yeah. And he says, but tell me what you're going to present. I said, OK. He says, don't go off script, and don't talk about giant celery or whatever you talked about at that last talk. So interestingly, Ken was able to herd all of us cats, and Ken got the grant, which actually shows there are second lives in academic life, particularly here in America. Well, going back to the end of the first act. So after licking our wounds, Tim Gardner, who was one of my START PhD students, he and I kind of sat back and said, OK, with more than 96 hours, what could we do in this space? And we began to read some of the books and talk to some of the leaders, Charles Lissy and others here at BU and around the country. And many were encouraging us to try to think about reverse engineering natural networks in cells. So by reverse engineering, it means to infer or figure out how are the different proteins and genes wired up inside a cell. It was an interesting challenge in about 97 or so because there was very little data available. And so Tim Gardner and I kind of considered the problem and ran as fast as we could away from that problem. And kind of sat back and said, OK, we're not going to reverse engineer, but we're engineers. Could we think about something else? And we realized, hey, why not take molecular components? So why not take genes and proteins and think about putting them together in circuits as an engineer would, like an electrical engineer, and build synthetic but wet circuits that could go into a cell where you knew or would try to design them to behave in the way you want. And this kind of helped launch the field of synthetic biology, where Tim and I kind of sat back and said, OK, let's come up with a schematic just as an electrical engineer or systems engineer might, do some mathematical modeling, and then consider getting it into a cell through DNA and see if it behaves in the way you want. So Tim and I sat back and said, OK, what could we design that would be complicated enough to be worthy of analysis, but simple enough that we could actually analyze it and maybe build it? And what we came up with was this notion of a genetic toggle switch. Okay? In short, here's a schematic for it. It's basically set up by two genes, repressor 1 and repressor 2. They're driven by promoters. These are on switches for genes. These on switches are set up so that they're always on. Okay, the system's designed that gene 1 wants to be on and gene 2 always wants to be on. Okay, you then set it up so that the protein produced by a gene, that's the product of a gene, wants to bind, say the protein produced by gene 1 wants to bind to the on switch for gene 2, shutting it off. And the protein produced by gene 2 wants to bind to the on switch for gene 1, shutting it off. Okay? 1 and 2 both want to be on, and they're each trying to shut each other off. Okay? For those of you who have siblings, you appreciate the human analog, right? In principle, you should be able to set this system up so that it can be bistable. That is, it wants to exist in one of two states. State 1, gene 1 is on and gene 2 is off. And state 2, gene 2 is on and gene 1 is off. Okay? In principle, you should be able to flip it between the states by transiently delivering an inducer for the currently active gene. That is, deliver a chemical or a stimulus that will shut off the active gene. Say, for example, you're in state 1. Gene 1 is on, gene 2 is off. Okay, if you deliver a chemical that can shut off gene 1, that will allow gene 2 to start producing its protein that's going to come now and shut off gene 1. And then you can remove your chemical and environmental stimulus, and you flip the state, the system, from state 1, gene 1 is on, gene 2 is off, to state 2, gene 2 is on, gene 1 is off. Okay, Tim and I got excited about this notion. We, again, went to do where we normally go. We did mathematical modeling. So here, I won't go through it in detail, but U is basically the concentration of the protein produced from gene 1. V is the concentration of the protein produced by gene 2. Alpha 1 and alpha 2 are effective synthesis rates, and you've got interesting coupling between these two. You can do different types of analyses that you might get as an undergrad here at BU. And we were able to actually do these analyses to show that the idea was not crazy, at least from a mathematical standpoint. That is, that if you properly balance your two genes, you could set this scheme up so that it could exist bistably. Now, state 1 is state 2. Here would be state 1, where gene 1 is on, gene 2 is off, and here's state 2, gene 2 is on, gene 1 is off. We had slews of these types of plots, all mathematical. And we decided, OK, again, like in stochastic resonance, we wouldn't have an impact if we limited ourselves just to the mathematics. So we started and went to talk to molecular cell biologists. And we talked to about 10 of them. And we brought in our transparent spots. Hey, what do you think? And invariably, the conversation almost always went as follows. Hey, what do you think? Oh, very interesting, guys. Really? Yeah, oh. Do you think it'll work? No. Why? Well, you see, it's going to leak. You're going to get the protein that should be off. It's going to be produced. It's going to sneak out. It's going to shut it. You're going to get this. The cell's not going to like it. It's going to shoot it out. You're not going to be able to, the cell's going to be upset. How are you going to do this? And invariably, they'd end the conversation by kind of patting Tim and myself on the head. Guys, biology's complicated. Well, we didn't get discouraged, and somehow we convinced Eric Eisenstadt, who was running a gene networks program at the Office of Naval Research, to give us $500,000 to try to build us an E. coli. What the ONR does, Eric's program, is that after you get the money, they have this program where all the people get money have to go and give a presentation. So we get invited to give the presentation. Katie, my daughter's about to be born, so I can't go, so I send Tim. Tim presents all the slides and gets torn to shreds by Nobel Prize winners, leading people, saying, this is crazy, it's not going to work. The next day, Eric Eisenstadt calls me, actually in the maternity ward, he says, Jim, Tim did not do well. You're going to have to, boost him up. Nobody in the room thinks it's going to work. I think it's going to work. I just don't think you two clowns are going to get it to work. I said, why? And he says, well, I know you're not a molecular biologist, you're a physicist turned bioengineer, but you didn't tell me Tim's. And I said, oh, I'm sorry, I forgot. I didn't mention that. You see, because Tim was a mechanical engineer out of Princeton. He used to design autonomous helicopters, and he flew them around the Princeton campus. Had no biology experience, and that came through in his talk. So Eric said, that's a lot of money I'm giving you. Go talk to somebody who knows what they're talking about, or I want my money back. And we went up to Charles Cantor. He says, Charles, what do you think? And he says, oh. And we explained. He says, crazy enough idea. I think it's going to work. 2, you two are so naive and experienced about how hard it's going to be, I bet you two could get it to work. True. And with that ringing endorsement, Charles opened up his lab to us. And what Tim did was he got in there and actually learned enough molecular biology and really used genetic engineering. So what you can do with genetic engineering is you can use different enzymes that can serve as scissors to cut out different genetic components, genes, promoters, other start-stop sites, and use other enzymes as glue to create different circuits, or in this case, plasmids, rings of DNA. Okay? And from that, we basically designed a toggle switch. We've got 2 genes, in this case LAC and let's go CI, that are both repressors, and we've got 2 promoters, PL and PTRIC. So the LAC repressor that's produced by this gene wants to bind to this promoter, shutting it off, and the CI gene produces the Lander repressor that wants to bind to the PL promoter, shutting it off. You can induce them. LAC can be induced by delivering a sugar, IPTG, and CI can be induced with heat by giving heat shock. You then want to report on it. You've got to figure out what state you're in. So what we use is a reporter protein called GFP. It's green fluorescent protein that was isolated out of jellyfish. For those of you who track the news, it was actually the focus of the most recent Nobel Prize in Chemistry that was given to the scientist that discovered and developed it as a research tool. Professor Shimomura, who's a chief scientist at Woods Hole Marine Biological Lab, is actually an adjunct professor of physiology here at BU. We're very proud of both his major accomplishments as a researcher, but also the honor of the Nobel Prize, which is really great. GFP also, I know there's some, how many comic book fans in the audience? I was a comic book fan. Don't be shy, you're among friends here. The popular culture of GFP is there's a new book out called The Science of Superheroes, where they actually purport that GFP accounts for why the Incredible Hulk is green. It purports that Bruce Banner was doing molecular biology with GFP when he was hit by the gamma rays and it got worked into his genome. That hasn't happened in my lab yet, although it could. What we then did was get this into E. coli, our favorite bacterium. This is not the nasty stuff that'll kill you in your hamburger. It's neutered, but you don't want to eat it. Nonetheless, you get sick. But you can get these into E. coli, and Tim was able to do that. And here's real data where he built 4 different versions of the toggle switch. So just to give you a feel for what we've got, he starts them in the off state. Okay? In this case, LAC is on, GFP and CI is off. Hits it with IPTG. After 6 hours, it flips basically nicely from the off state to the on state. You can remove IPTG, and it now stays in that on state for as long as you want to look. E. coli divide about every 20 minutes. We've looked out 96 hours, and they stay in the on state provided it's bistable. You can then whack it with heat, change in temperature, and flip it from the on state to the off state. And similarly, it will stay in the off state for as long as you want to leave it. With this system, you can actually then characterize really sharp switching thresholds. So as you increase the amount of chemical that you might want to detect, the system will stay in the off state till some critical concentration whoop, flips up. Okay, which is important for biosensing applications, which I'll mention in a couple minutes. You can also characterize how long does it take this thing to flip, to switch. For this switch, for the molecular biologists in the audience, we actually had it on a high copy plasmid. So in any given E. coli, there are about 80 to 100 copies of the switch. So going from the off to the on state, it actually took a long time, about 6 hours, for this thing to flip. On the other hand, going from now the on to the off, where you're just going to do heat shock, it only takes about 20 or 25 minutes, where you're basically just waiting for the protein to basically destabilize and fall off the DNA, allowing then transcription and translation to come on. So what do we do here? Well, it's nicely captured in an ad from Accenture that came out a few months after we published this. Bacteria tested as digital circuit. Now it gets interesting. It's exactly what we did. That is, a genetic toggle switch was basically modeled on this notion of a toggle switch or flip-flop that is at the heart of digital computers. So it's an RS latch. It's this very standard switch that you have in electrical engineering. We thought about use that as motivation and introduce basically a biological version of it, one that had memory just as an RS latch or a toggle in electronic form would. With that, you think about open up possibilities, maybe biocomputing. For those of you who read Scientific American or Discover or American Scientist, popular science, you'll know there's a lot of interest in biocomputing in this country and has been for about a decade. DNA-based computing, cell-based computing. When we presented this the first time, which was 1999, at an amorphous kind of biocomputing workshop, I was giving the talk, went through the slides, and the room was like, yay, the BU guys have done it. Woohoo, biocomputing's here, we have a talk, woo. I'm like, okay, wait a second, guys. And I put up that last switch. It says it takes 6 hours for this thing to flip. All the air went out of the room. Why? Well, the computers that you have at home or the computers you have in your little machines you carry around, the switches in those systems take tiny fractions of seconds to flip. This thing takes 6 hours. We can get it done in 20 minutes if you have a single copy. That's still a long time, right? I don't think anytime soon we're going to have a PC with E. coli inside and a little label sitting on your desk. Imagine surfing the web in that case, right? We all get frustrated, right? If the network's a little slow, the switch is slow. Now check, see, what's the weather going to be tomorrow, right? Well, 2 days later, on your E. coli computer, oh, it was supposed to rain yesterday. I didn't realize that. Not going to happen, right? As much as there's a lot of talk about it, there's a lot of attractive things. These things self-replicate, they self-repair. Boy, there'll be great capability in computers. It's not going to happen for many reasons, and I know there's questions after which I can elaborate on. That's the wrong way to think about biocomputing for these circuits. The right way is if you think about now using these engineered circuits as ways to produce programmable cells. That is, can you take these engineered devices and interface them with both natural and other engineered systems in cells. Cells have been evolving for millions, billions of years. They have great capacity to sense different things. They have great capacity to do funky things in the environment. So we got excited about doing this with Hideki Kobayashi and Mads Kern, Michi Araki in our lab, of taking our toggle switch in particular as a little interface to go in. And one of the first things we thought about doing was designing it as a biosensor. So there is increasing interest as a result of bioterror, as a result of environmental remediation, in designing cell-based sensors that you can put in the environment to detect different things, whether it be a toxin, a pathogen, an attack, some nasty chemical. What's interesting about E. coli and other bacteria is they have an SOS system. So an SOS system is a DNA repair system. If the DNA is damaged, this system gets flipped on. Okay, so we decided, okay, let's start easy. Can we couple our genetic toggle switch to the DNA repair system to be a DNA damage sensor? That is, could we detect something in the environment that's damaging? Couple it in, this is a nice funky circuit. Basically, we got this thing to work beautifully. One of the things you want is you don't want your system to have false positives. Right? If you're going to use it to detect something, you don't want it going off when the thing you're trying to detect is not there. Right? Imagine how crazy you are if your smoke alarm is going off in your home and there's no smoke or no fire. Right? So similarly, from a biosensor, you don't want it flipping into the on state when there's nothing there. And this thing is incredibly stable. You sit it out in the environment for 2 days, it hasn't flipped. But if you deliver mitomycin C, which is a nasty chemotherapy agent that damages DNA, you can detect very, very tiny amounts, 1 nanogram per milliliter, with the switch flipping from the off state into the on state. And as you increase the amount of mitomycin C, you detect more and more of cells flipping on. You can also detect UV. For the sun gods and goddesses in the audience, it damages your DNA. If you deliver UV to your cells, it's going to flip on your switch, and even very, very tiny amounts of UV get detected by the cell. In this case, we're using GFP as an output, so it's nice fluorescent. Another output you could use is biofilms. Biofilms are communities of bacteria that are encapsulated in an extracellular polysaccharide matrix and attached to a surface. Plaque on your teeth is a biofilm. The gunk in your bathrooms are biofilms. The stuff on the side of ships are biofilms. We'll come back to that in 2 slides. But it's a really powerful phenotype that you can see. It's very easy to see. And so what we did was took out GFP and added a gene that if flipped on will produce biofilm. I won't show you the detailed results, but is now tested with our DNA damage sensor. No DNA damage present, boom, you deliver DNA damage and you get nice biofilm, there's a readout. So you can now maybe just fly over a field that you're trying to detect some scheme and actually see, there was a detection event. And what's nice about the toggle and also nice about the biofilm is with memory, you don't want to always be kind of standing over your sensor like this. Okay, nope, nothing, right? You want to be able to inquire about it every once in a while. And the toggle switch has a memory to it that you can actually do that to pick up, okay, we're fine. Oh, now you go back and check every 24 hours something happened. What you can do with this is my colleague Ron Weiss at Princeton actually went further and actually engineered it further so that you could actually have bullseyes formed. So that you could use fluorescing bacteria, biofilm bacteria, to actually identify the actual center of a chemical spot or of some scheme you wanted to detect. Turns out, again, as I said, bacteria have a lot of interesting potential and different capacities, including the ability to detect heavy metals in the environment. So you can imagine now using these sensors to detect lead paint on toys coming in from overseas, or lead paint in a new house, or finding mines and minefields. One of the biggest problems we have around the globe. You have these minefields that have been left. So if you could actually use microbes to detect, there it is, without losing a person trying to find it, which you could use with these bullseyes and these textures would be great. While we were doing this, we actually got approached also by the biotech industry, Dow and DuPont. What they do at Dow and DuPont is they basically use microbes to produce proteins of interest. How do they do it? They engineer them to produce a protein. They then grow them up in these huge bioreactor vats, up to a high density. When they get to a high density, they throw on a bunch of chemicals, flip them on then to produce the protein. This is happening now also in the bioenergy space, and I'll speak very briefly about it in a couple slides, where people are now using microbes to convert biomass into some energy source. The big challenge in all of these is it costs a lot of money in these bioreactors to grow them up, and then the chemicals are a pretty big cost. If we're going to move towards this as being a real feasible option, you've got to get the price down. So these guys came to us and says, hey, could you use synthetic biology to program these guys to flip themselves on when they hit a certain density? And in short, what we did was actually take advantage of the fact that bacteria can actually talk to each other. They actually have different quorum sensing systems where they can sense, okay, how many of similar bacteria to me are in my environment? We took that system, engineered it, coupled it to our toggle switch, and used it then to flip on switches that could produce proteins once they reached a certain density. And it actually worked just as we expected, which is nice. And actually now we're talking, and Dow and DuPont are looking about incorporating into different bioreactors. One of the challenges I said with bioreactors and earlier of other schemes is you'll actually build up biofilms on the outside. They're nasty. You have to use big boatloads of chemicals to get them off the side of your bioreactors. That's a problem in itself. The bigger problem is that they are a major health problem. Why? Basically, bacteria and biofilm are highly resistant to antibiotics. They're about 1,000 times more resistant than free-swimming bacteria. It's actually how bacteria generally exists is in biofilms. So now if you have a medical implant, either a cardiac device or, say, an artificial hip, right, that the big risk now is not the surgery, it's the infection you might get on your implant. Charles DeLisi, our dean, actually had a major infection on his artificial hip. The poor guy had to go in, as well as David Wallace, pitching coach, who did not coach me for the throw. But at the Red Sox, who 2 years ago, David Wallace actually had to go in. They had to remove his hip, replace it, but in fact, in the meantime, allowed the infection to clear. Huge problem and very nasty. You can't get after these things. So with Tim Liu and Hideki Kobayashi, Mads Kern, we actually decided as bioengineers, could we actually re-engineer something to actually go after these biofilms? And what we went after were bacteriophage. Bacteriophage are viruses specifically designed or that basically go after bacteria. So how do they work? This little kind of lunar module of a bacteriophage will dock onto a bacteria and blow its DNA into the bacteria. Okay, then it can follow one of two paths. It can either be a nice house guest, where it kind of tucks in the DNA, goes to sleep, doesn't do anything. Or it can be a bad house guest, like college students, where what do? They eat up all your resources, make copies of themselves, and explode your house. Woo! And what happens then is these phage will reinfect the bacteria and do the same thing again if they're in this lytic phase. Trouble is, in biofilms, they can't get below that initial surface. Remember, these things are in kind of a gunk, right? So with Hideki Kobayashi, what we did was basically re-engineer the phage so that they would express on their surface an enzyme that would degrade the biofilms. So we had here was basically a two-prong attack. They would both be lytic, and they would enter and destroy the biofilm matrix, right? So Keanu Reeves-type biofilm busters were designed to go in. And in short, these things work brilliantly. So I won't go into details, but basically here's biofilm gunk on a plate. We got brilliantly clear when we actually delivered these viruses designed to attack the bacteria. And shown here, we basically can effectively eliminate the bacteria biofilm. Tim Liu's gone further and basically now re-engineer these guys to be kind of Trojan horse sentinels. They go in, and instead of expressing the enzyme on their surface, they actually sneak in and produce proteins inside the cell that will break up the biofilm. And he's actually got similarly brilliant work. Now, all this sounds pretty cool, nice on a Tuesday night. Not everybody reacts so positively to some of the things we're doing. There is a lot of and growing concern in the community that synthetic biologists are out of control, and that people like me will design something that will be really nasty, escape from my lab, and eat Fenway Park. That's not going to happen. In interest of time, I won't go now, but if somebody wants to ask, I'll talk about it. To guard against this, what we've thought about instead was, could you program these cells that might go in the environment as detectors so that they would self-destruct after a certain period of time? And so with Ari Friedland, what Ari did was take RNA switches that were developed by Farron Isaacs and Dan Dwyer in our lab and design genetic counters. So these were counters that could be designed so that after the cell divides n number of times, it kills itself. So you have kind of an expiration period of your guys so that they do get out, or if you have them out intentionally, they can't last very long. So you basically set it up as a cascade. I won't go through it, but it's basically primed from the beginning to go. To detect one event, it flips on and produces some proteins that triggers you to go downstream to produce another set of proteins after the second detection event. So this system was designed to count to 2. And Ari actually got this thing to really work beautifully in E. coli so that it needed 2 detection events to actually flip on. It's linearly extendable so that you can actually add components to count to 3, 4, 5, 6, 7. Here it's counting to 3 by same scheme, and he similarly got this thing to work really beautifully. Where are we going with this on synthetic biology? There are many applications. We've moved from bacteria into yeast. We moved from yeast into mammalian cells. One of the big applications for the toggle switch in the mammalian space will be cell and gene therapy. This general medical technology is coming and will have an impact on many of the people's lives in this room. The notion on gene therapy is you're going to introduce genes into a person who has a defective gene or missing a gene and having as a result of disease through a viral vector or some other scheme. In cell therapy, you'll remove cells from a person, re-engineer them maybe to produce insulin, and reintroduce them into the body. Once we figure out how to do that safely without getting an immune response, the next challenge will be how do you actually control the gene of interest so that it's on when you want it and it's off when you want it. In our business, you can get catalogs for gene switches. Just as the L. Albini catalog, you order a sweater or a nice new Parker or a nice shirt like I have tonight. We have catalogs. You can get switches. You can buy already precious switches that are genetic switches. What are those? These are as if I walked in this room and to keep the lights on, I always have to have my finger on the switch. For genetic cases, that you always have to have a chemical present. The toggle switch is different, right? The toggle switch is like the light switches you have here. You walk in, you flip it once, it's on. And then you can wait as long as you want, and then when we're done, they're going to flip the switch off, the lights go off. For the toggle switch, the nice thing from a therapeutic standpoint is the patient would only have to take the pill once to flip it on. And then if they're having a bad reaction, take it once, flip it off. Well, when Tim and I went to talk about this in the early 2000s, about 8 years ago, to pharmaceutical companies, said, guys, what do you think? Isn't this great? And I said, they only have to take one pill. Yeah, you know where this is going. So they would say, OK, guys, Tim and Jim, what's your business model? What do you mean? And we sell pills, guys, is what they, and they say, call a taxi, get Jim and Tim out here. True stories. That aside, given we probably can't get any financial backing for this, is that the real advantage will be as a safety switch, right? So from an FDA standpoint, as you move forward, imagine now getting these therapeutic schemes into the body. You can't really get them out. So now imagine you have a bad reaction, and now having to take a pill all the time to keep something off that should be on to help you, but you have to keep it off because of a bad reaction. Toggle switch you could have is you flip it on, keep it off. I mentioned earlier the biosensors, bioenergy. We're working with some talented guys in our lab, Kevin Latovsky, Moronas, who are now engineering bacteria in kind of the bioengineering space to actually produce bioplastics, degradable plastics, which we probably report on the next year. And Les Flash Gordon, we're actually very interested in functional genomics, coming back to this notion of reverse engineering. So about 6, 7 years ago, Tim and I kind of walked around. We had a hammer, these synthetic gene networks. Where were nails that we could hit on? And now about 4 or 5 years from when Charles and Stu Kaufman and Leon Glass encouraged us to think about reverse engineering natural networks, we got re-excited about it because there were many more data sets now available. So on the heels of the Human Genome Project, we now have these long parts lists, genes, proteins, promoters, parts of DNA. But we don't have instruction manuals. That is, we don't know how these parts are all fitting together to produce underlying biology. Okay? And that's going to be really the next biology and next bioengineering of the next decades, if not this century. Tim and I got excited, realized, hey, we could use these switches to go in and probe natural networks and figure out how they're wired up, just as an electrical engineer would. So if you're an electrical engineering student, you'll invariably take a lab course or have a professor who will hand you a circuit board and say, OK, hotshot, how's this wired up? So what do? Well, you go off to the lab, you flip different things on and off in the electronics lab, measure as many things as you can, and you come back to the professor and say, hey, there you go. And nowadays, given grade inflation, no matter what you show them, they give you an A. But you still show them, say, hey, this is what I found, right? In kind of how many of you can relate, it might instead be that if you buy a home and you don't have the wiring diagram set up of your home, but you have a circuit breaker box in the basement, what do? You take your boom box, turn the volume on, plug it in the kitchen, run downstairs, you flip the switches till you hear the boom box go up kitchen, okay? Run back upstairs, plug it in the dining room, run downstairs, flip the switch, dining room, right? You do that, you get your first order map of your house. Then you can do other perturbations to see, are they connected, right? This may be determined, it might be intentional, it might be just random, right? So Katie and Danny might be making microwave popcorn in the kitchen. I might be in my study with the air conditioning on, Mary's in her study with the space heater on, boom! Circuit blows, and you say, okay, those 3 rooms are connected. And from that, you infer the system. Well, with those networks, we then thought, okay, what can you do with these? Right? We get quantitative networks as an engineer would. And we realized a big application was that we could use these networks to figure out what drugs were hitting. What happens is that biotech pharmaceutical companies are really good right now at setting up chemical assays to tell you what is a drug under development, is it hitting what it was designed to hit? Right? So from a target base, you'll say, I really want to nail down this protein to get the change in disease. They're good at doing that. What they're not good at doing is telling you what else is that thing hitting. In the case of antibiotics, you have about 4,000 possibilities. Antifungals, 6,000 possibilities. Humans, 30,000 possibilities. And it's why, if you own biotech stocks, it goes red a lot. Why? Phase 2 trials fail, phase 3 trials fail, because they hit a lot of other stuff. We realized that we could use these networks to basically identify what the drugs are hitting. I teach control theory. There are many of my students here. From the very first lecture, and including even on April Fool's Day lecture, I'll always basically say there are 3 components to a control system, right? The input, the output, and the system itself. If you know 2 of the 3, you can work out the 3rd. We actually work out the network by knowing our inputs and measuring our outputs. From that, we can infer the network. Now, if you've got a drug and you don't know what it's hitting, but you have your network, and you can measure the response of all the proteins or genes in that cell, you can then use the network, filter the output through the network to predict what's your drug hitting. Neat idea that we actually showed works nicely in bacteria, fungus, and mammalian cells. And what we got really excited about and focused on in the last 2 years is using this now to identify what are antibiotics hitting and what are they stimulating in bacteria. Why? Well, it's captured nicely in this slide. Basically, we have resistance is going through the roof. Increasingly, there are resistant strains out there. They're originally in the hospitals, now out in the community. So this supply is going up, and unfortunately, the number of new antibiotics being developed and approved is going down. So it's a scary situation. There are nasty bugs out there. They're going to get us if we don't actually change this around by figuring out getting new insights into how can we kill off the nasty bacteria. What we did was turn to our network. So here's just a tiny part of the network inside E. coli. Developed off of hundreds of measurements of genes and proteins in those by Jeremiah Faith and Boris Hayed as part of our group. And what Dan Dwyer and Mike Ohansky did was actually use those networks to look at quinolones. Quinolones are a class of antibiotics that basically damage DNA. Cipro. You hear about Cipro all the time. Some of you probably had Cipro, some of you may be on Cipro. Damages DNA. That's the way it's generally thought to kill the bugs. Dan and Mike actually used our network approach to actually identify a completely novel pathway where it leads to the production of hydroxyradicals inside the bacteria. Hydroxyradicals are oxidants. So many of you take antioxidants, you eat a lot of blueberries, and you worry it damages your cells. Well, the oxidants also damage bacteria. So the hydroxyradicals will damage DNA, lipids, proteins, leading to cell death. Mike Ohansky kind of sat back and said, okay, that's interesting. The quinolones do that. I wonder if other antibiotics do the same. So within the cytolytic antibiotics, you have major classes. You have quinolones, as I said, that damage DNA. You have beta-lactams, like ampicillin, that block cell wall synthesis. And you have aminoglycosides, like gentamicin, that mess up protein translation. They have very different targets, and the thought was that those different targets led to different cell death pathways that killed the bacteria. Mike, in a brilliant insight, says, oh, I wonder, studying what he and Dan had done, I wonder if, in fact, cytolytic antibiotics all stimulate hydroxyradicals. And, in fact, that's what they found. They found that basically if you deliver these antibiotics, boom, the hydroxyradicals are formed. And they actually worked out this pathway through a network analysis that in all cases, all these different antibiotics all have a common pathway that leads to the production of hydroxyradicals that contribute to death. Cool biological and clinical insight, but the real significance of this is that Mike and Dan were able to then go show that if you knock out the DNA damage repair system, right? So the hydroxyradicals will damage DNA. the cell will flip on DNA damage repair system and try to protect itself and kind of get back to healthy. If you block that, you can basically make all cytolytic antibiotics more effective. So if you come up with a small molecule, you can make super cipro, or super ampicillin, super gentamicin, super penicillin. So what we did with Guillaume Cotterell was actually both use a BU resource that's supported by the NIH, Center for Clinical or Chemical Methodology and Library Development. It has about 2,000 compounds. And we also used a biodefense facility at Harvard Med that had 50,000 bioactive compounds. We screened all of them and found several that could actually shut off the SOS system. One of which actually was FDA approved. Interestingly, this is a toxic drug. I don't know why the FDA approved it, but it's a toxic drug. Turns out it's a natural antibiotic that's used for cancer. Again, not sure on how that made it through. But when you deliver these drugs along with the cytolytic antibiotics, you make them more effective. Shown here is gentamicin, an aminoglycoside. When you deliver it with our little molecule, boom, you make it 1,000 times more effective. And we're now working to actually move this out of the lab, out into commercial space, and considering it as small molecules. Really neat idea. Came from Mike and Dan. Again, the cytolytic antibiotics will produce hydroxyradicals that will help kill. Well, then sat back, and Mike Bohansky thought, OK, hydroxyradicals, there's the beneficial. There's also a really concerning aspect of hydroxyradicals, and that is they damage DNA, and they can lead to mutations, OK, that maybe could lead to resistance. So Mike first wanted to see, does it actually lead to an increase in mutation rate when you deliver cytolytic antibiotics? And that's what he found, well, pretty scarily. So when you looked even at very low levels of antibiotics, ones that aren't killing the cells, you get a bump up in the mutation rate. And this bump up is directly related to hydroxyradicals that are being produced. Mike then speculated and said, OK, if you're getting these mutations, well, generally you'll have resistance between bacteria that's formed. Either they kind of send each other the resistant bacteria, they exchange it, that resistant genes that they developed, or you'll have the antibiotic basically kill everything off except for a few that naturally develop the mutation that has made it protective against that antibiotic. Mike actually said, OK, if you're getting these high mutation rates, I wonder if you're getting broad emergence of resistance among these bugs. And this is one of the scariest slides, if not the scariest slide I've ever put up. This is exactly what he found. So delivering low-level ampicillin, what he found was even just after 5 days that you develop resistance not just to ampicillin, which is shown here in green, but to all other cytolytic antibiotics, some even more than to ampicillin. So if you have the sniffles, or you feel a little run down, don't bother Dr. Mary or your primary care for antibiotics. It's leading to nasty bugs out there that are going to get us. Okay? And it's this really scary notion, and there are other ways that we're now thinking about how we might be able to shut this down. Where are we going? Well, in our lab, we're actually looking at these persistent infections. We're moving into pathogens, looking at MRSA, looking now at TB. There's a big effort with the NEDL, the National Emerging Infectious Disease Lab, with Mark Klempner, who's here, and James Galligan, a new hire, to apply these network analyses to TB. We've not limited ourselves just to bacteria. We're looking at mammalian diseases. We're now using our network approaches to identify what are the genes that are being turned up or down that lead to cancer, brain cancer, breast cancer, lung cancer, liver cancer. We're looking at neurodegenerative diseases, ALS, Parkinson's, using similar network approaches. And probably most exciting, we're actually using the networks to identify combination therapies. And that is, most drugs to date are being developed where we're going to try to knock down a single gene or protein. But it's crazy. It's if you're going to kind of put your finger in a dike to hold back this huge thing where you've got 100 things that need fingers in. What we're doing is using a network approach to actually identify multiple things you can knock down, whether with drugs or with RNAi, from a network biology standpoint to develop new therapies. So where are we? You know, there's a great slide from AT&T years ago. Is networking in your genes? Years ago, it was not for me. By strange turn of events, with given this great environment here at BU, it was, and our group is now born to network. And with that, just cues me up for a few special thanks. I want to start off just to thank the BU community, Bob, David, Andre, Ken, Saul, Charles, Nancy, for really creating and sustaining an interdisciplinary environment here at Boston University that's not only supported but really celebrated. Also, I want to thank you for putting up with me for all these years. I've been here since Michael Dukakis was governor, which is really quite a long time ago. I want to thank the members of my lab, a great group of young people. I'm incredibly proud of all that they've been able to accomplish, and I'm really thankful that they're so willing to take on highly risky projects that have almost no chance of success. And they come in every day, and they're willing to take those risks, which is tough to do. It really is. When you want to get your degree, you want to finish. And I appreciate they're willing to do that, and I appreciate they're willing to tell me that my ideas are wrong-headed and bad, really on a daily basis. And I appreciate they seem to take pleasure in telling me that. To my friends, many of whom are here, the St. Ege group, the Bowen group, Nashua, Holy Cross, I want to thank you all for just being so much fun You know, there are a lot of young people in this audience thinking about becoming scientists. Most days, most weeks, your experiments don't work. Most of your good ideas turn out to be bad. Don't limit yourself only to always work hard, be in the lab, work hard, but don't limit yourself to the lab. Make sure you have a great group of friends who are a lot of fun, or you're going to be miserable and cranky. Just as important, a great group of friends, you need to have a great family. I've had the luck of the Irish to have a great Irish family. To my siblings, a couple over here, Kathleen and Kevin, to my cousins, my aunt and uncle, I'll get to you too, to my in-laws, aunts and uncles, cousins, I want to thank you guys for creating a competitive, challenging environment that's really made academia seem tame. And if you don't believe me, come see us at the holidays. To my folks, Jim and Eileen, they're here, which is great. Special thanks for always having education as a priority in our home. My mom insisted on us kids that we work hard, that we were grounded, stay grounded, and that we were humble. My dad, on the other hand, encouraged us to have our head in the clouds, to try to be one of the best at whatever we chose to be and not to be shy about it. And as a result, I'm incredibly conflicted with many issues. And then finally, Mary, Katie, and Danny, one couldn't ask for a better family. These three make everything fun and interesting. I think they could actually make a faculty meeting fun and interesting. Mary, as many of you know, is a primary care physician at Mass General and Harvard. She's taught me many aspects of medicine through the years and introduced me to fascinating medical problems. And she's had the good sense to remind me on really a daily basis that even though she's teaching me all this medicine, I am not a medical doctor and should not offer advice. And Katie and Danny have inspired both Mary and me and challenged us to really stay sharp at home. Each night it's as if Mary and I are contestants in Are You Smarter Than a First Grader? Are You Smarter Than a Fourth Grader? And we've yet to win an episode, which bodes well for the future. Thank you. And
I know Jim is willing to take a few questions. Are there some from the audience? We have mics set up on both sides.
Hey, Jim, that's a fantastic talk. Thanks a lot. One thing that I thought of when I was hearing the first part of your talk, where you're talking about the noise, I was wondering, are there any natural systems that use added noise to increase their sensitivity? Any biological systems that use that trick?
yeah, so we actually discovered that the proprioceptive system uses noise in that way. So the proprioceptive system is your sense of limb position in space. Right? So if you close your eyes, this isn't a sobriety test, but if you close your eyes, put your hands out, and actually try to touch your nose firstly, but actually then try to touch your fingertips in the middle. It's quite hard to do. And that is, you do it as a result of your proprioceptive system. And what happens there is your main sensors, as my control students will know from lecture 1 of the class, are spindles. So you have little spindles in your muscles that sense stretch. Well, those are driven or respond to alpha motor neurons that go to your skeletal muscles, but they themselves have little muscles inside them, gamma motor systems. And we did work with Tim Inglis from UBC to actually show that the gamma motor system serves as a natural noise source to tune up your spindles. So for example, the case would be if you're going to try to thread a needle. Right? Probably the better way is you stay calm, you reduce your muscle, and you put it through. The way I do is you're like this, right? And when you're doing that, is you basically, in some way, are actually increasing your sensitivity for slight movements. And so we were able to show that it's a natural noise source, one such source.
Quick question, 2 questions. First one, what is it called? What's the concept whereby you develop something in the lab, but you don't really understand what's going to happen when it enters out into the world? Is that like the cascade effect or something? The second question, what do you think of the do-it-yourself biology movement, establishing protocols for students and enthusiasts to basically run like electrophoresis experiments in their garages or kitchens or what have you?
Yeah, so first I would call that dangerous. And it might be emergent behavior, which is popular among several physicists who are on my right here, going back 10 years ago. I'm not sure. I expect it's probably emergent or due to the complexity of the system. You can't necessarily predict how it's going to behave. In terms of do-it-yourself, there were a few pieces in the Globe I saw within the last couple months. This is this notion that a new movement to kind of as you had with electronics and kind of computers in the 70s to kind of move molecular biology so that kids could do stuff at home. I think in general it's a great idea. The more the kids could be exposed at an early age and have kind of the freedom to create. I think it's going to be a bit different than the computer side because molecular biology is really expensive. So my lab loves to give me purchase orders that I need to sign, and it runs to the tens of thousands on a monthly basis of the types of supplies we need, let alone the equipment. There's millions of dollars of equipment in our lab. So I think to do the interesting things, it's not going to be feasible. To do the small things, I think it's a great idea. Thank you. Sure. There was a question. Oh.
Yes.
Have there been any experience on putting, supplying a stochastic signal to the central nervous system, supratentorial, to see if that affects either psychiatry or more central motor or sensory function?
So there's been some studies where they've applied noise to increase attentiveness and overall activity within central neural systems. In terms of psychiatry, I haven't seen studies where they intentionally say did the direct application. There's certainly, and there are many parents in this room, there's the anecdotal that we've all experienced where you'll have the fan going on in the back or the wave machine that has the random white noise to actually calm down your little kid, to get the kid to sleep, or let alone before we had the nice noise cancellation headphones for airplanes, you would actually have white noise devices that at least would serve to calm. In terms of the detailed psychiatric studies, I haven't seen any. We've actually been approached by a number of autistic groups that wanted to see, could you use the technology to help as therapeutic interventions for the autistic kids. We don't work on kids in the lab, but we've directed them toward groups that might.
Yes,
Yeah.
thank you.
So how far are we away from bacteria to lead up oil spills?
There are bacteria already that can eat up oil spills. So there's some that have that capability. The challenge, as with most of this, is it's one thing to have a cool university lab demo. The other is to have something that's robust enough to be in the environment. And it's one of the comments that I said I returned to potentially in questions is that most of the stuff we work on is exceptionally weak and has not been designed to live for more than a few minutes if it got outside my lab on Cummington Street. So that going back, it's one of the earlier applications in early genetic engineering in the 80s. There were groups that actually found bacteria that could eat up oil spills. The challenge is, can you now have these so that they won't get out of control when they're out there? Will they actually exist nicely with all the other nasty things that would, say, be in the ocean if you put it in the ocean? But we have those capabilities. It's just has somebody made the decision to actually re-engineer them to be as robust as they need to be effective. Danny.
How do the bugs break the house?
Oh, how do the bugs break the house? So the bugs, good question. So they break the cell by just making a lot of copies of themselves. So they made multiple copies and they just exploded the house. So if you come to our lab, you can actually watch these things exploding cells. They pop, the cells pop, and the lunar guys trickle off to go get another cell. So they use the DNA and the protein machinery of the cell to make many, many copies of themselves. And so it's overcrowding. Good question.
Other questions? Wait,
Oh.
yes. Mr. One? All the way down,
Oh, yeah.
yes.
Hi. He's one of my former students. He did well in the class. Just so you know.
So in differential equations, we study if you excite a system, you periodically with the sine wave, you could cause it to go into resonance and just blow up, right? So suppose instead of noise, if you were to excite a system with, say, a periodic chemical stimulus, could you cause it to go into, say, chaos and use that to study maybe chaotic phenomena that can enhance?
So it's a really interesting question. So Leon Glass, in the early 80s, who's a pioneer in knowledge dynamics and was one of my mentors when I first came here to BU, actually did studies on chick cells, where he did that. He delivered a periodic signal, ramped up both the amplitude and frequency, and was able to shift the cells into cardiac chaos in the chick case, and actually study the phenomenon. So chaos in this case is not, as we vernacularly call it, kind of crazy disorder. It's actually seeming disorder in a deterministic or set-up system. And so you could get that. The challenge at the cell level, so there it's a large cell. Challenge at the cell level, it's really noisy. and it sounds like I talk a lot about noise, but there's so many fluctuations that I've not been able to see a protocol where you could, say, periodically drive a cell, that you could reasonably see chaotic behavior versus just wild, crazy, noisy behavior as a result of the stimulation. Go here first.
Yes.
Do you think that these interesting results would have any implication for better understanding the nucleus of life?
The question is, do any of the results that I present give us insights into the emergence of life? And I've not been directed to going after that problem. It might be due to my religious upbringing, that I already have enough challenges with relatives in my family that if I now say I'm going to try to create life, they already think I have a God complex. But that aside, that we, at the RNA level, so there's a lot of interesting work being done in this city and around of looking at how could life have arisen on the result of different chemical soups. And there's increasing movements toward the RNA world. That is, that they're viewing RNA as kind of an early molecule because one of the tricks is how can you get replication is one of the key. And Farron Isaacs, who's here, and Dan Dwyer, who's here, who did work early on these RNA synthetic biology, I think it may speak to some sense of what's kind of the simplest circuits or simple schemes that could be self-replicating. But we haven't been directed toward that. It's a good question.
I think with that, I will cut the questions off. We have two mementos for Jim for tonight. One is more permanent probably than the other. The first is a plaque with his poster, and second is a check that if he gives his broker may not be permanent.
My broker's here. It's my brother.
Congratulations.